Good evening, everybody. In this class, we are going to learn AVERY, MACLEOD, and MCCARTY transformation experiment. Actually, this transformation experiment is further proves the justification of DNA as a genetic material. This transformation is the experiment is the continuation of Griffith transformation experiment. Here, actually, the Griffith transformation experiment leaves a question: what is the transforming principle which converts R strain to a strain of Streptococcus pneumonia bacteria? Now, let us going to study regarding this. What is the aim of this AVERY, MACLEOD, and MCCARTY transformation experiment is to identify the nature of transforming substance responsible for converting non-virulent R strain to virulent A strain. How live R strain is converted to S strain. What is the transforming principle that we are going to study? Here, heat-killed S cell were taken and DNA, protein, RNA and proteins were isolated, which are thought to be any one of these as a genetic material from the S cell. Lipids and sugars are removed. Here, three test tube all DNA, the solutions of DNA, RNA and proteins were taken in three test tube, and first test tube we need to add proteinase, and next the RNAs were added, and the third one the DNase enzyme were added. What is the purpose of adding this enzyme? Proteinase, which acts on protein which are present in the solution, it digests the protein, hence there is no protein in this first tube. Then second tube RNAs, which acts upon RNA, and the RNA were digested, there is no RNA. And DNase, the enzyme which acts upon DNA, then DNA were digested and there is no DNA. So these solutions were taken in conical flask, and finally the R cell were added. If there is no protein, so R cell were added and finally results in appearance of S cell, that means the virulent S cell. So if protein is responsible for protein as a genetic material, means there will be no S cell here, but S cell appear. So it concludes that protein is not a genetic material. Then in this second solution, the RNA, there is no RNA in this solution, R cell were added and S cell also appear here. That means if RNA as a genetic material means there is no RNA, then S cell would not appear here. So here this second conical the experiment conclude that RNA is not a genetic material. Then third test tube, there is no DNA which is digested by DNase enzyme, and this solution R cell were added, but there is no appearance of S cell. That means what is the conclusion here? So if the DNA is a genetic material, means there is no DNA, there is no formation of S cell. Suppose if DNA is present, means there will be a formation of conversion of R cell into S cell. So S cell would appear here. So this experiment concludes that DNA is one of the main transforming principle which transforms R cell into S cell. So no DNA, no S cells. Okay, so this is what AVERY, MACLEOD and MCCARTY experiment what deals about. Now I hope you understood about the logic of this experiment and how they arrived the conclusion that DNA as a genetic material. Here only it was there. So add to culture containing living R cell, observe the transformation by testing for the presence of virulent S cell. This three test tube is for this step. Then here treat solution to enzyme to destroy protein, RNA or DNA. These enzymes were added. Second step. First step, all these are removed, only the DNA, RNA and the proteins were isolated. So this is what about this experiment. Further we need authentication that DNA as a genetic material. So this experiment, earlier the scientists were not convinced that by proving the DNA as a genetic material by this experiment. So next experiment for further final authentication that DNA as a genetic material is Hershey-Chase-Martha experiment that we are going to study in next class. Okay, thank you. Have a nice day.